But part 4 for the chapter 26, we're going to look at transcription and translation. So here you have a DNA that stays in the nucleus of the cell, never leaves the nucleus. So it needs to be transcribed. And the strand that is transcribed is known as the template strand. The complementary of that original helix is the informational strand, not going to be used. The mRNA or messenger RNA molecule is the exact RNA duplicate of the DNA informational strand, with the exception that the uracil replaces thymine that you find in the DNA strand. So this is here a summary. Looking at just the nucleus of the cell, you see the double helix of the DNA, and that's going to lead to transcription to messenger RNA. So look at the example below. You have two strands, DNA, informational, and template. So informational runs from 5' to 3'. The template is the opposite, 3' to 5'. And the messenger RNA is a template strand, which is pretty much the same as the informational strand because it's the mirror image where T is going to be linked with A, and A, well, in the informational strand will be linked to T, but remember there's no T-thymine in RNA, there is instead uracil. So ATG, that was in the informational strand, complementary template strand TAC, messenger RNA is AUG. And then template strand GGT, we give you CCA. And look, template strand runs from 3' to 5', so messenger RNA is going to be the reverse, 5' to 3'. So how does it start? Well, RNA polymerase recognize a control segment in the DNA that precedes the nucleotide to be transcribed. This is the start codon. And we're going to look at the genetic code. This is made of triplets of consecutive bases. That's what we call codons. Nucleotide triplets carried by the messenger RNA code for one specific amino acid that are going to be linked or assembled one after the other into the protein. The sequence of those nucleic acid code is going to be the complete protein, and that's what we call a gene. The RNA polymerase moves down the DNA segment, adding complementary nucleotide to the growing messenger RNA strands. And then that transcription ends when the RNA polymerase reaches a sequence known as termination sequence that tells that's the end of the sequence that needs to be copied. So looking here, a good summary, double helix of DNA, looking at a specific location such as the genes were here. You look here on the number 2, the template strand. The other one is the informational strand. Only the template strand is going to be used to make that messenger RNA, which is the one you have on the very far right, mRNA strand. At the end of the transcription, the messenger RNA molecule contains a matching base for every base that was on the informational strand, except that uracil replaced thymine. But you need to be aware that about only 10% of the base pairs in DNA code for something or genes. The one that code for a gene is contained in one or more small section we call exon. The one that are coded for nothing, non-coded sequence, we call those introns. So looking here at the summary, the initial messenger RNA, which is not ready to be sent out, contains introns and exons. Only the exons that are in orange are going to be used. So introns are excised, removed, with the help of what we call a spliceosome. So to memorize, what goes out exits the nucleus are exons. The introns are eventually spliced out. The ribonucleotide sequence in a messenger RNA chain is like a coded sequence, and I will give you the order in which amino acid residues should be joined together to make the protein. And those are new definition. The codon is the sequence of 3 ribonucleotides in the messenger RNA chain that code for a specific amino acid. And then the genetic code is that sequence of nucleotides that are coded in triplets. We call them the codons. We find them in messenger RNA, mRNA, and that determine the sequence of amino acid, the primary structure in the proteins. Now, looking at the genetic code, there are 64 possible 3 bases combinations in RNA. And actually 61 code for a specific amino acid and 3 code for chain termination, the stop codons. The meaning of each codon is the genetic code. And we also have a start codon, AUG, that codes for methionine. Codons are always written in the 5' to 3' direction. Know how to read here that table. The first base on the 5' end, so uracil, cytosine, adenine, guanine. Second base where you have for each of them is your uracil, cytosine, adenine, guanine. And then the third base on the 3' end is also uracil, cytosine, adenine, guanine. And if you read this, which I wish was expressed better in that table, the first base I read U, second base U, third base U. So U will code for Phe for phenylalanine. U will code for serine. UCC also code for serine. UCA also codes for serine. So that's the triplets. Now, if you look at this, you see that you have different combination of the triplets that will code for the same amino acid. However, you will never have two amino acid for the same triplet. So that's where the genetic code is very specific. Another thing that I wish they said better in that textbook is the fact that the genetic code is universal. That means that UUU coding for phenylalanine, Phe, here, that's what we find for humans, cats, dogs, corn, every organism that follows the genetic code, so that is universal. Finally, I would like you to notice that the 5' end of the messenger RNA strand codes for the N-terminal amino acid. So here summary, DNA informational strand, where the one that is used is the template strand. From there, 3' to 5', you have the messenger RNA that runs from opposite 5' to 3', which basically is the same as the informational strand, except that you replace T-thymine by uracil. And then it shows you on the bottom each triplet that codes for different amino acid making that protein. So AUG is actually the starting point methionine, proline, valine, glycine, histidine, leucine, and serine.